Okay, so now let's talk about the Calvin cycle. We just got done talking about the light capturing reaction that produced ATP and NADPH that are now going to be used in the Calvin cycle. In addition, the Calvin cycle is going to use carbon dioxide from cellular respiration, particularly from the citric acid cycle. But the goal of the Calvin cycle, in addition to regenerating these products that are going to be needed for the light capturing reactions, the main goal here is going to be to make sugars. And to do that, the Calvin cycle has a fixation phase, a reduction phase, and a regeneration phase. The fixation phase, as we'll see in a moment, involves using carbon dioxide and fixing it to molecules in the Calvin cycle. The reduction phase is the part that's going to reduce the total number of carbons in the Calvin cycle, but this is when sugar intermediates are made. And then regeneration regenerates the molecule that carbon dioxide fixes. By regenerating this molecule, the Calvin cycle can continue. But let's break this down in a little bit more detail. Just to remind you, as we get moving here, the Calvin cycle occurs in the chloroplast. But unlike the light reactions that occur in the thylakoid membranes, the Calvin cycle, or stage 2 of photosynthesis, you could call it either, occurs here in the stroma. So everything we talk about here is in the stroma. So let's begin with this first stage. This first stage, remember, which is the fixation. Fixation involves carbon dioxide, in fact 3 carbon dioxides, entering the cycle. And it enters the cycle by adding this carbon to a molecule that we're going to call RUBP. RUBP has 5 carbons. And we're going to be adding a carbon to each of them, so we're going to have 3 RUBPs. Using this enzyme called Rubisco, and you need to know this enzyme. This is in my top 3 favorite enzymes. And we'll talk a little bit more about Rubisco in a moment. But it's going to use Rubisco to attach these carbons to these RUBP molecules. Or what we say, it's going to fix it to it. And in doing so, it's going to generate 6 3-PGA molecules. Now each of these 3-PGA molecules are composed of 3 carbons. So this ends the first step, the fixation of these carbons from the carbon dioxides to the RUBP molecules to generate PGA molecules. Now the 3-PGA molecules are going to be involved in this next phase of the Calvin cycle. And this phase, remember, was called the reduction phase. And it's going to make 6 molecules that we're going to call G3P. Now this is where the reduction part of the reduction phase comes in. One of these 6 will be pulled out of the cycle. So one G3P molecule is pulled out, and through various other reactions will make glucose and other macromolecules. So we've reduced the total number here. And I should also point out that each of these G3P molecules also contain 3 carbons. And we just lost these 3. You might have caught on then that the way we're going to get those 3 back is through these 3 carbon dioxides that enter. But we'll come back to that in a moment. At this point, we've ended the reduction phase. Next, we can talk about the regeneration phase, our third phase of the Calvin cycle. As we enter the regeneration phase, remember, we no longer have 6 3-GP. We only have 5 of those molecules because we lost one during this reduction phase. So we only have 5 of these, still each with 3 carbons. So during the regeneration phase, these 5,3-GP molecules have their bonds reorganized to generate 3 RUBPs, each with 5 carbons. So we're making a larger molecule, but the total number of carbons is the same. Here you have 5 of these molecules, each of them containing 3 carbons. Here you have 3 of these molecules, each of them containing 5 carbons. So still 15 total carbons here, 15 total carbons here. So it's just a reorganization of the bonds to regenerate RUBP. Now something that might be bugging you is that I told you that ATP and NADPH that were generated in the light reactions are necessary for the Calvin cycle here. But I haven't talked about them yet. So let me tell you where they feed in at. They feed in here, where we use 6 ATP and generate 6 ADP, plus the inorganic phosphate. These are now taken back up and can be used in the light reactions. Also, in this reduction phase, 6 NADPH molecules are used and converted into 6 NADP plus molecules. And then over here, we're going to use some more ATP. We use 3 ATP, and it produces 3 ADP and 3 inorganic phosphates. We require this energy of ATP at both of these steps because both of these steps are breaking bonds and reorganizing the chemical bonds in these structures. So using fixation, we fix carbon to this RUBP molecule to make this molecule here, the 3-PGA. It then undergoes a bond rearrangement using ATP and NADPH to generate 6 3-GP molecules. During the reduction phase here, which is here, we lose one of those G3P molecules, which is converted later on into glucose and other molecules. The 5 remaining G3Ps have their chemical bonds restructured, reorganized, and we generate 3 RUBPs so that we can continue the cycle over and over again. Now I want to say something about Rubisco here. As I said, now I want to talk about Rubisco not just because it's one of my favorite enzymes, but because it's an incredibly important enzyme. If nothing else, it just makes sugar. And I don't mean just make sugar so food tastes good, but this sugar will be the building blocks for many, if not most, large structures within the plant cell, and ultimately in the cells of organisms that eat the plants. So it's not just sugar like we might think of it. It's these building blocks that are really important in all of life. An interesting thing about it is that it is a very slow enzyme, dreadfully slow. In fact, this reaction here from RUBP to 3-PGA is not an energetically favorable reaction. But what makes it favorable is that we're constantly producing the reactants here of RUBP, but also it's very abundant. It makes up about 50% of chloroplast proteins. If you recall, all the different enzymes and proteins that are necessary in the light reaction, in the Calvin cycle, not to mention all the other processes that are occurring in the chloroplasts. This one enzyme makes up about 50% of it. That's one way that we can keep this reaction going. It is also, I guess I'll have to write this over here, sorry for the sloppiness here, is it's the most abundant protein on Earth. If you think about all the plants and all the chloroplasts and the fact that it's 50% of all chloroplast proteins, there's no other protein that is more abundant. So if I ever ask you, what's one of my most favorite enzymes? Rubisco. Now that we've finished the Calvin cycle, there are a few other things I want to talk about before we end this chapter on photosynthesis. And what I'd like to do is draw some plant cells here. We talked about the photosynthesis here and how within these plant cells, oxygen is produced and it is released, and that CO2 from the environment has to enter In addition, water can also be lost. So how do these molecules enter and leave the plant cell? They enter and leave the plant cell through structures called stomata. So there are stomata all over the leaves. Now the stomatas aren't always open because there are times when you'd want to have CO2 come in and times when you wouldn't want CO2 to come in. Same with oxygen, same with water. It needs to be regulated at some level. And it's regulated by the presence of these things called guard cells. Guard cells open and close the stomata. Now remember that plants aren't just one cell thick or one cell type. So we may have multiple layers of plant cells. And the CO2 concentrations are not going to be as high in these deeper layers of plant cells. But the Calvin cycle must still occur in those cells. So what's going to happen here? And I'm going to kind of make this cell here bigger just so we can see it when I'm going to explain to you. This cell here, which is receiving higher concentrations of CO2, can be converted in some plants into a 4-carbon molecule. Remember, what I just told you was that CO2, when it enters the cell, it enters the Calvin cycle and it generates a 3-carbon molecule. This is generating a 4-carbon molecule. Plants that can do this we call C4 plants because they can generate this 4-carbon molecule. Now how is that going to help the cell below it? Well, there are openings in these cells. They're not stomata. Stomata are on the outside. So these openings are called plasmodesmata. We're going to learn this in the next chapter. So if you learn it now, it's just one less thing you have to learn in the next chapter. But these plasmodesmata are channels between different cells. And these 4-carbon molecules that are generated can now enter this cell. Once in this cell, it can create 3-carbon molecule. But the best thing that it does, for the plant anyhow, is it generates CO2 or releases CO2. This CO2 can now be taken to the Calvin cycle, which I'll just abbreviate as CC. So what this acts as, these 4-carbon molecules that are produced abundantly on the surface here, they act as carbon reservoirs that can now be taken and deliver CO2 to the parts of the plants that have a low concentration of CO2. Now I want to talk about just one more thing, and I promise this is the last thing. Okay, so this last thing I want to talk about are desert plants. Plants that live in this hot and dry environment. Now usually a plant cell during the daytime would have their stomata open so they can bring in CO2 so that they can use that CO2 during photosynthesis. However, if you're in a hot and dry place, you want to keep those stomata closed. Those guard cells are keep it closed so that water isn't lost and so the plants don't dehydrate. The drawback to that is during the daytime, carbon dioxide cannot get into the cell. Yet, we know the Calvin cycle and photosynthesis occurs in these desert plants. So how do they accomplish this? So what happens is at night, when the sun has gone down, these stomata open. They don't have to worry about losing too much water, even though they could because all of the stomata open. But what happens now is a rush of CO2 enters into the cell. Now at this point it's useless to the cell because there's no light, so you're not generating that NADPH and the ATP from the light reactions. So the CO2 is just sitting here. And what happens is it's stored into these molecules that we're just going to call CAM. C-A-M. And CAM will hold on to these carbons, and then when the sun comes out, again, the stomata close, the CO2 can't get in, but that's okay because there's this big store of carbon in the plant cell that can now be used in the Calvin cycle. So this is a clever way that these desert plants have adapted to survive in these hot and dry places by storing carbon at nighttime so they don't dehydrate. Okay, that's all I want to say about the Calvin cycle and this last part of photosynthesis. If you have any questions at all, please make sure you come see me. If not, I'll see you in class. Bye.